During the Second World War, naval sonar operators kept detecting something that made absolutely no sense according to the instruments they were using. Sonar works by sending out a pulse of sound and measuring how long it takes to bounce back off a solid surface, which lets operators calculate distance to the seafloor or to a submerged object like an enemy submarine. But operators across multiple navies kept picking up a false seafloor, a layer that reflected sound as if it were solid ground sitting at a depth of a few hundred meters, considerably shallower than the actual ocean bottom beneath it. Stranger still, this false floor moved. It rose toward the surface at night and sank back down during the day, tracking a daily rhythm that no static seafloor feature could possibly follow. Some operators initially wondered whether they were detecting some kind of enemy technology or an unusual geological formation, but the daily vertical migration ruled out anything geological almost immediately, since rock formations don't rise and fall on a 24-hour cycle in sync with sunlight. This phenomenon became known as the deep scattering layer, and for decades after the war, it remained one of oceanography's most persistent and genuinely strange unresolved mysteries, hiding in plain sight, detected by nearly every sonar-equipped vessel that ever passed over open ocean, and yet never fully explained or measured with any real precision. It has taken until remarkably recently, using technology and research methods that didn't exist even 20 years ago, for scientists to finally solve this mystery with the kind of confidence that allows for a real answer rather than an educated guess. And the answer turns out to matter enormously for understanding how the entire planet's climate system functions. The basic explanation for what causes the deep scattering layer was worked out relatively early, within a couple of decades. After the war ended, once marine biologists started comparing sonar readings against net trawls and direct observation, the layer turned out to be biological rather than geological, made up of enormous numbers of small fish, squid, and other organisms living in the ocean's twilight zone, the region between roughly 200 and 1,000 meters deep, where sunlight still penetrates faintly but not nearly enough to support meaningful photosynthesis. These organisms, dominated by small, deep sea fish called bristlemouths, along with various species of lanternfish, squid, and crustaceans, spend daylight hours in the darker depths of the twilight zone, avoiding visual predators that hunt using whatever faint light filters down from above, and then migrate upward each night toward the surface, following the setting sun into shallower water where food, in the form of plankton, becomes more abundant near the surface after dark. This daily journey, repeated by an almost incomprehensible number of individual organisms simultaneously, is now recognized as the single largest synchronized animal migration on Earth, dwarfing anything that happens on land, including the migrations of wildebeest across the Serengeti or caribou across the Arctic tundra, simply because of the sheer number of individual organisms participating in it every single day, in every ocean basin on the planet, without exception. Knowing that the deep scattering layer was biological explained the mystery in a basic conceptual sense, but it left an enormous practical question completely unanswered for decades afterward. How much life, in terms of actual measurable biomass, does this layer actually contain? This question turned out to be extraordinarily difficult to answer using the tools available for most of the 20th century, because the standard method marine biologists use to estimate fish populations, trawling nets behind research vessels, and counting what came up, systematically failed to capture these organisms accurately. Many twilight zone fish, particularly bristlemouths and lanternfish, are small, fast, and highly effective at avoiding slow-moving trawl nets, meaning net-based surveys likely captured only a small fraction of what actually lived at that depth, understating the true population by an unknown but potentially enormous margin. For decades, oceanographers had a rough, low-confidence estimate suggesting mesopelagic fish biomass, the scientific term for organisms living in this twilight zone, totaled somewhere in the range of 1 billion metric tons globally, a number that was already considered enormous, exceeding the combined weight of every other type of wild fish in the ocean. But researchers themselves were the first to admit this figure rested on shaky methodology, since it was built primarily from trawl data known to systematically undercount the very organisms it was trying to measure. The breakthrough that finally allowed scientists to solve this problem came from combining acoustic survey technology with a very different approach to sampling, one that treated the sonar data itself as the primary source of information, rather than relying mainly on physical net captures to validate what the sound waves were detecting. A landmark study published in 2014 led by researchers, including Xavier Irigoyen and a team working with the Malaspina expedition, a round-the-world oceanographic research voyage specifically designed to study the deep ocean's biological and chemical properties, used advanced acoustic surveying equipment capable of distinguishing between different types of biological signals in the water column with far greater precision than earlier sonar technology allowed. Rather than relying primarily on trawl nets, which the research team used mainly to help calibrate what different acoustic signatures actually represented biologically, the study used the acoustic data itself, gathered continuously across the ship's entire round-the-world route, to estimate total biomass across ocean basins directly from how much sound the water column reflected back at depths corresponding to the twilight zone. This approach produced a result that stunned much of the oceanographic community. Rather than roughly 1 billion metric tons of mesopelagic fish biomass, the corrected estimate came out somewhere between 10 and 30 times higher, placing global mesopelagic fish biomass potentially as high as 10 billion metric tons, a figure that, if accurate, would mean these small, largely unstudied twilight zone fish alone outweigh essentially all other wild fish biomass in the entire ocean combined by a considerable margin. The scale of that number is worth sitting with for a moment, because it represents such a fundamental revision to how scientists understood the basic structure of ocean life. For context, total global wild fisheries catch, representing the entire commercial fishing industry's haul across every ocean on the planet in a typical year, sits at roughly 80 to 90 million metric tons annually. The corrected mesopelagic biomass estimate suggests a single, largely uncaught, and until recently poorly understood group of small fish contains roughly 100 times more total biomass than the entire global fishing industry manages to catch in a year across every targeted species combined, including tuna, cod, herring, and every other commercially significant fish population currently supporting the world's seafood supply. This means that for most of the history of marine science, researchers had been building their understanding of ocean biomass and ocean food webs around a picture that dramatically underestimated one of its single largest components, simply because the primary tool used to measure it, trawl nets, happened to be poorly suited to catching the very organisms in question, creating a systematic blind spot that persisted for the better part of a century before acoustic methods finally corrected it. Solving the biomass mystery mattered scientifically in its own right. But it mattered even more once researchers started working through the implications for a completely different area of Earth science, the global carbon cycle and the ocean's role in regulating atmospheric carbon dioxide. This connection exists because of the twilight zone fish's daily migration pattern, the same behavior that originally created the deep, scattering layer mystery back during the war. When these fish rise toward the surface each night to feed on plankton, and then descend back into the twilight zone each morning to avoid predators and bright light, they aren't just moving themselves vertically through the water column. They're also transporting carbon, because the plankton they consume near the surface contains carbon absorbed from the atmosphere through the ocean's surface layer, primarily via photosynthesis performed by phytoplankton. And once that carbon becomes incorporated into a mesopelagic fish's body through feeding, it travels downward with the fish as it migrates back to depth, where a portion gets released through respiration, waste, and eventual death, at depths where that carbon becomes effectively isolated from the atmosphere for potentially centuries, sometimes considerably longer, depending on ocean circulation patterns in that particular basin. This process, known to oceanographers as the biological pump, was already recognized as an important mechanism for ocean carbon sequestration before this mesopelagic biomass revision. But the earlier understanding of biological pump strength was built on the older, dramatically underestimated fish biomass figures. Once researchers recalculated the biological pump's efficiency using the corrected mesopelagic fish biomass numbers, accounting for how much carbon these fish likely transport through their daily migration, given their now much larger estimated population, the resulting figures suggested that mesopelagic fish migration might account for a considerably larger share of the ocean's total carbon sequestration capacity than climate models had previously assumed, potentially representing the transport of 1 billion metric tons or more of carbon into deep water annually through this migration mechanism alone. Given that oceans already absorb roughly a quarter of all human carbon dioxide emissions released into the atmosphere each year, any significant revision to how efficiently the ocean sequesters that carbon into deep water, where it stays isolated from the atmosphere rather than potentially cycling back to the surface and re-entering the atmosphere over shorter timescales, has direct relevance for climate models, trying to predict how quickly atmospheric carbon dioxide concentrations will rise, given any particular level of human emissions. This is part of why marine scientists describe the mesopelagic biomass discovery as solving one of the ocean's biggest mysteries, rather than simply an interesting technical correction to a fish population estimate. The revised numbers don't just change how biologists categorize fish population statistics in an academic sense removed from practical consequences. They potentially change how accurately climate scientists can model the pace of climate change itself, because a stronger, more efficient biological carbon pump than previously assumed means the ocean may currently be doing more work absorbing and sequestering human carbon emissions than climate models had been crediting it for. which matters enormously for understanding how much atmospheric carbon dioxide concentrations would be expected to rise under any given emissions scenario, and consequently, for predicting the pace and severity of resulting climate impacts like sea level rise, ocean acidification, and shifting weather patterns that depend heavily on how much carbon actually accumulates in the atmosphere rather than getting sequestered in the deep ocean. The commercial and conservation implications of this discovery add another dimension to why it matters, and this is where the story becomes considerably more complicated and, in some ways, more urgent. As global fish stocks in traditional commercial fisheries have faced mounting pressure from overfishing, habitat degradation, and climate-related shifts in ocean temperature and chemistry, some fishing industry stakeholders and policymakers have looked at the newly revised mesopelagic biomass estimates and seen an enormous, largely untapped potential food resource, a reservoir of fish biomass potentially large enough to significantly expand global seafood production if commercial fishing techniques could be developed to harvest these small, deep-dwelling fish efficiently. Bristlemouths and lanternfish, while extremely abundant, are small, oily, and not naturally suited to direct human consumption in the way that larger, more familiar fish species are, but they could potentially be processed into fish meal, fish oil, or other food and feed products if harvested at industrial scale, similar to how anchovies and other small forage fish are currently processed for aquaculture feed and other industrial uses. Given the enormous estimated biomass involved, even harvesting what might seem like a small percentage of the total mesopelagic fish population could represent a fishing operation larger than most of today's existing commercial fisheries combined. This commercial potential has alarmed many marine scientists and conservation researchers precisely because of the carbon sequestration connection just described, creating a genuine tension between two different ways of viewing the same newly discovered resource. If mesopelagic fish populations play as significant a role in ocean carbon sequestration as the revised biological pump calculations suggest, then large-scale, meaningfully weaken one of the ocean's most important natural mechanisms for offsetting human carbon emissions, potentially accelerating the very climate change that increased fishing pressure elsewhere, is already partly a symptom of, since warming oceans and shifting fish stocks are themselves driving fishing industries to look toward previously untapped resources like the mesopelagic zone in the first place. Removing a substantial fraction of the fish responsible for transporting surface carbon into deep water through their nightly migration could, in theory, reduce how much carbon the biological pump moves out of atmospheric circulation each year, creating a feedback loop where addressing pressure on traditional fisheries by developing new ones in the twilight zone risks undermining the ocean's broader capacity to regulate atmospheric carbon dioxide, a trade-off that wasn't even part of the conversation before scientists solved the biomass mystery and revealed both how large this fish population actually is and how significant a climate role it likely plays. This tension has led marine policy researchers and international bodies to push for caution before any large-scale mesopelagic fishing industry develops, arguing that decisions about whether and how to harvest this resource need to account for its climate-regulating function, rather than treating it purely as an untapped protein source waiting to be exploited, using the same approach that has historically been applied to traditional fisheries, often with limited consideration for ecosystem-wide consequences until after overfishing had already caused serious damage. Some researchers have specifically warned that repeating historical patterns from other fisheries, where commercial harvesting expanded rapidly before adequate scientific understanding of sustainable yield limits and ecosystem impacts had been established, could be particularly consequential in this case, given how directly mesopelagic fish biomass appears to connect to global carbon cycling, rather than simply representing an isolated food web within a specific ocean region. This has resulted in growing calls within international fisheries governance discussions for a precautionary approach to any future mesopelagic fishery development, prioritizing further research into sustainable harvest limits and ecosystem impacts before any significant commercial expansion into this newly quantified resource takes place, a markedly different posture than how many traditional fisheries developed historically, where commercial exploitation often significantly outpaced scientific understanding of sustainable limits. Beyond the carbon cycle and fisheries policy implications, solving the deep scattering layer mystery has also reshaped scientific understanding of predator behavior throughout the ocean, because an enormous, newly properly quantified food source doesn't exist in isolation from everything that depends on it. Many species that were already known to feed at least partly on mesopelagic organisms, including various whale species, seabirds, tuna, and sharks, turn out to depend on this food source more heavily than earlier. Lower biomass estimates would have suggested was even possible, given how much total prey biomass those estimates implied was available. Research into deep-diving whale species, for instance, has increasingly focused on how closely their foraging behavior tracks the vertical migration pattern of mesopelagic fish and squid, with some whale species apparently timing their own dive patterns to intercept these prey populations as they migrate through different depths over the course of each day and night, suggesting an evolutionary relationship between whale foraging strategy and mesopelagic migration patterns that developed over a very long time scale, shaped by a food resource considerably larger and more reliable than scientists previously understood it to be. The technological story behind finally solving this mystery is itself worth appreciating, because it illustrates a pattern that keeps recurring throughout ocean science more broadly. Mysteries that persist for decades often aren't due to a lack of scientific interest or effort, but due to a genuine mismatch between available technology and what a particular question actually requires to answer properly. The deep scattering layer had been detected by sonar since the 1940s, correctly identified as biological in nature by the 1950s and 60s, and yet its true scale remained essentially unmeasurable with any real confidence until acoustic technology developed the precision to distinguish between different biological signal types within the water column, and until research expeditions like Malaspina were specifically designed to gather acoustic data continuously across enough of the world's oceans to support a genuinely global biomass estimate, rather than isolated regional measurements that couldn't necessarily be extrapolated with confidence to the ocean as a whole. This same pattern, a phenomenon detected and roughly understood for decades before the specific tools needed to properly quantify it finally became available, recurs throughout deep ocean research. And it's part of why oceanographers are often reluctant to describe any particular ocean mystery as permanently unsolvable, since history suggests that persistent mysteries tend to eventually yield once the right combination of technology and research design gets applied to them, even after remaining stubbornly unresolved for the better part of a century. Research, since the original 2014 biomass revision, has continued refining these estimates, using additional acoustic survey expeditions, improved calibration methods comparing acoustic signals against a wider variety of net sampling techniques designed specifically to better capture fast swimming, mesopelagic species, and computer modeling that accounts for regional variation in mesopelagic biomass across different ocean basins rather than assuming a uniform global average. Some of this follow-up research has moderated the most extreme estimates somewhat, suggesting the true figure likely sits toward the lower end of the original 10 to 30 times revision rather than at its absolute upper bound, while still confirming that mesopelagic fish biomass substantially exceeds the older, pre-2014 estimates by a wide and scientifically significant margin, regardless of exactly where within that revised range the true figure ultimately settles. This kind of gradual refinement, where an initial breakthrough finding gets progressively narrowed and clarified through subsequent research rather than either fully confirmed or overturned, represents a fairly normal and healthy pattern in how scientific understanding develops after a major initial discovery, and it doesn't diminish the significance of the original finding so much as it reflects the ordinary process of science converging toward increasingly confident and precise answers over time. Standing back from the specific numbers involved, what makes this story compelling as an example of a genuinely solved ocean mystery is how completely it reframes something that had literally been detected by human technology for over 70 years, before anyone could properly explain its true scale and significance. Sonar operators during the Second World War were, without realizing it, detecting the single largest synchronized animal migration on the planet, a migration involving potentially 10 billion metric tons of fish moving vertically through the water column every single day, playing a significant and only recently appreciated role in how much carbon dioxide the ocean removes from the atmosphere each year, a process directly relevant to how quickly human-caused climate change unfolds. That such an enormous, planet-spanning biological and climatic process could go essentially unmeasured and poorly understood for the better part of a century, hiding in data collected continuously by naval sonar operators who had no idea what they were actually detecting, says something important about how much of the ocean's fundamental workings remain hidden, not because they're inherently unknowable, but simply because properly understanding them requires exactly the right combination of technology, expedition design, and scientific attention, a combination that, in this particular case, took decades longer to assemble than anyone initially expected, and which continues to be refined even now as scientists work to translate this newly solved biomass mystery into policy decisions about fishing, carbon accounting, and ocean conservation that will shape how this newly understood resource gets managed going forward.